Welcome to Ask a Protein Pro. In this video, we will discuss tips for preparing samples and labeling peptides with tandem mass tags, or TMTs, for mass spectrometry analysis. Let's get started. Sample preparation encompasses a wide range of techniques including lysate preparation, protein enrichment, protein digestion, and sample cleanup. Proper sample prep is critical for mass spec analysis because the quality and reproducibility of the samples directly impact the results from mass spec instruments. By utilizing a sample preparation kit designed for mass spectrometry, you can save time and increase reproducibility. Thermo Scientific EasyPep MS sample prep workflow enables efficient and reproducible processing of cultured mammalian cells, tissues, and biofluids for proteomic mass spec analysis. The kit contains pre-formulated reagents which help provide an easy, optimized protocol that trims the time to generate MS-compatible peptide samples to less than 3 hours. This is a great time savings compared to a traditional workflow of over 30 hours. Depending on the amount of starting sample, different formats are available to process samples from 1 microgram to 2 milligrams, which results in high-yield, mass spec-ready peptides for downstream applications. Next, let's look at some steps necessary when preparing different types of samples with the EasyPep MS kit workflow before TMT labeling. Plasma samples must first be depleted of high-concentration proteins, such as albumin or immunoglobulins, that obscure detection of low-abundance proteins. Plasma samples require a longer digestion period of 2 to 3 hours to achieve less than 10% missed cleavages. Here are a few tips to keep in mind when preparing tissue for mass spec. Some tissue samples may require longer homogenization in lysis buffer. As an example, heart tissue is tough and dense and would require more time in the lysis buffer. Be sure to centrifuge following homogenization to remove any insoluble debris. Here are some tips when preparing formalin-fixed paraffin-embedded samples, or FFPE samples, prior to TMT labeling. Assess the quality of the FFPE block by H&E stains. To obtain sufficient yield of protein to label, be sure to use at least 6 micron thickness curls from FFPE blocks and at least 3 curls. Homogenize the samples in EasyPep lysis buffer after paraffin removal and rehydration steps. Now that we have discussed how to prepare samples with the EasyPep kit, we can label our samples with TMT reagents. TMT reagent sets can be used to label up to 18 different peptide samples. Normalize the sample amount before TMT reagent labeling by using a protein and peptide assay. This is helpful to label the same amount of peptides across different samples. TMT and TMT Pro reagents are highly sensitive to moisture. To avoid moisture condensation onto the product, equilibrate the vial to room temperature before opening. All amine-containing buffers and additives must be removed before digestion and labeling to ensure reactivity with lysine residues and peptide N-termini. I hope these tips for preparing samples for TMT labeling have been helpful. Visit our Mass Spec Resource Center and check our TMT webinars for more tips. Thanks for watching.